So welcome, everyone. Thanks for coming out. It's tempting to be outside, taking advantage of these great last summer-like days, but you're here. Thank you for that. We're actually celebrating the 10th anniversary of the 3 Minute Thesis. Yeah. The 3MT at UK. The 3MT, as you can tell here, has been around since 2008, and we brought it to UK in 2013. So we're glad to be able to celebrate the research across campus, but not only that, every time we have a 3 Minute Thesis, we're also celebrating the courage that it takes to do this. I mean, think about, and by the way, I'm going to introduce our dean in just a moment, and he's going to give his own 3 Minute Thesis, right? I gave you a warning about this earlier today, right? But think about what it takes to get up here and talk about your research to a public audience. Now, the students and postdocs competing tonight are very used to talking with their peers, with specialists at conferences, in classes, in the hallways in their departments. It's a very different thing to talk to a novice audience, and that's what the challenge is. So they have 3 minutes to do it in one slide. The trick is, and you'll see them doing this tonight, is to tell a story about their research. And by that we mean they're trying to engage you, they're trying to appeal to your interest, common ground, they're trying to hook you, pull you into the story, and lead you somewhere. And so that's what they're going to do. Promise you're going to do that, right? You all are going to do that tonight. Now they're revising it in their head now, and I've got them confused. Now, we're looking forward to all of your presentations. So that's also what we're celebrating, this courage that it takes to get up on stage, get out of your comfort zone, talk to a group of people who know very little about your research. That's something to celebrate. So give them another round of applause just for that. So the 3MT has gone through quite a bit of growth since 2013. It started out with just a handful of competitors that really just auditioned for the 3 Minute Thesis competition, and then we also coached them quite a bit, and we brought back the coaching this time. And actually, our 3 Minute Thesis coach is standing right there. Wave to everyone, Patty. Yep. So the finalists this time had the opportunity to get some coaching after they learned of their that they would be advancing, and most of them took advantage of that. So again, welcome. I'm not your official welcomer. I'm just giving you some pre-words, but let me introduce our new Dean of the Graduate School, Dr. Padraig Kinney. Dr. Kinney.
Thank you. It's super great to be here. This is only my, I think, 6th week in the graduate school and at UK, and I'm really proud to be here. And thank you, Morris and Ashley and others for all the organizing. So, you know, I think there is no greater favor that we can do a fellow scholar, and we're all scholars in this room, than to listen intently as they tell about their research. I mean, we've all been in those situations at a party or something like that, and someone says, "So what do you do?" Right? And you start kind of explaining your research, and you're feeling kind of weird about it because you're like, I know they're not really interested. And you can kind of see that very often, and look, my research is super interesting. We'll just stipulate that. But we've all been in a situation, I sure have, where I'm also feeling that they're kind of slipping away and thinking they really should go get another drink, but now they've asked somebody about their research. But honestly, that is the one thing we most desire, to be able to talk about our research and have others listen. You're all here to do that, and I really want to honor that. Those of you who are presenting, those of you who are not, you're all going to be listening to what others have to say and giving them what they really, really need. and you're going to be listening to the best of it. You're going to be listening to people really telling great stories about their research from areas that are so far from your areas of interest, but it's going to be great. And then the other thing that's wonderful about it is not only do you listen to their research, but unlike what would happen at your usual party, is you get to cheer and clap afterwards. That would be super strange at the usual party. Really let loose, tell them how much you enjoy their work, because you know that you would like the same. Okay, and without further ado, let me hand it off to Ashley Sorrell, who will tell you what's going to be happening next. Thank you all.
Thank you, Dean Kinney. So I'm going to give you a little bit of a background of GradResearch Live and what you can expect from tonight. We have essentially 3 tracks. One is the Pre-3MT research showcase, and this is for folks who are graduate students who are early in their research who may not have results yet. The second is the official 3-minute thesis competition, the one that Morris discussed coming out of the University of Queensland. And finally, we have the postdoctoral research showcase for postdoctoral scholars to present their research as well in a way for a lay audience. So there are some rules that our contestants must follow. You will notice there'll be a single static slide that each researcher will present. There can't be additional electronic media or props. Presentations, 3 minutes maximum. So we actually have someone down front running a timer as our presenters are giving their talks. Spoken word only, even though I would love to see interpretive dance with research. For this purpose, it is spoken word. So when a presenter starts the presentation through speech, I think the movement, that would kind of be hard to tell, but through speech, then the timer will start. And we have our judges down here. The decisions of the judges is final. Speaking of our judges, we have Chelsea Brislin, who's the Associate Director of the UK Gaines Center for the Humanities. Victor Puente, he's a morning news anchor on WKYT. And Rebecca Thomas, Dean of Faculty at Transylvania University. So they will be judging in all 3 tracks this evening, and we thank them for their time. So just like in a movie theater, we ask that you please silence your cell phones as to not interrupt the presenters as they're giving their talks. All right, so we are going to start with the Pre-3MT showcase. And first, we have Hannah Blevins from Entomology, The Complexities of Cockroach Control.
If I were to ask by a show of hands, how many people in this room have encountered a cockroach in your home or business, I doubt I would see very many hands raised. But this is not because cockroach infestations are rare. On the contrary, cockroaches are one of the most prevalent and pervasive indoor pests in the world. This is due to their variety of adaptations, such as their affinity for a wide array of food sources, like the crumbs you left on your counter this morning, and their ability to squeeze into small cracks and crevices, successfully evading us. Despite their prevalence, cockroaches have become associated in our culture with uncleanliness, and this has led to feelings of shame and embarrassment that have prevented people from getting help for their cockroach infestations. Cockroach infestations not only negatively impact us mentally, but also physically. For example, cockroaches produce metabolites in their bodies that lead to exacerbation of allergy symptoms. If left to exposed long-term, these compounds can lead to the development of asthma, which is a chronic inflammatory lung disease. This is of particular concern as the CDC currently reports that around 24 million people in the US are currently dealing with asthma, and around $50 billion are spent in the US annually on asthma-related healthcare. What's worse is that these effects are felt most strongly by members of our communities who are socioeconomically disparaged. Although we understand the importance of managing cockroaches for the health of our community, there are more than just emotional barriers that are preventing people from getting help. Although it is currently understood that pesticide application remains the most effective means of controlling indoor cockroach infestations, professional treatments can be costly. And DIY over-the-counter products have a history of being misused and overused, both of which have led to the development of resistance. Resistance has left many cockroaches to be resistant against our pesticides, and this is where my research comes in. I'm currently working with multiple populations of German cockroaches, the most common cockroach species in the US, and I'm looking at how resistant these cockroaches are to a variety of active ingredients of many different pesticides, both for home use and commercial use. I'm also looking at what it is within the bodies of these cockroaches that are leading to this resistance, and how we as a scientific community can share this knowledge with our pest control specialists so that we can work together to mitigate these effects. This is the main goal of my research. I want to help make cockroach control more affordable, effective, targeted, and safe for the betterment of the health of our communities. Thank you.
I should mention that between contestants, we are going to have some time for the judges to fill out their scores. So there will be some silence during tonight's event. I did not make any jokes or anything like that. All right. So next in our pre-3MT track, we have Devin Donegan, who will be telling us about inefficient broadband and how it hinders academic performance and economic development. Devin?
Take a moment to think about a time when you had little to no access to broadband infrastructure to complete something that was urgent at the time. Did you grow frustration? Did you have to relocate to a local library or even to a family or friend's house? Well, people in low-income urban areas and people in rural areas are facing major technical communication policies in these areas, which is causing low-income areas to be affected socioeconomically and social mobility, which is a ripple effect in this case where it is hindering people academically and economically. This ripple effect causes people to cause an issue for food deserts, increase in low-income areas with unemployment, and also hinders low-income people as students in these areas to be have low test scores. When you have low test scores in these areas during the 2019 to 2020 pandemic, causes people to suffer tremendous because of the brightness of people have to social distance. When people have to social distance, it's caused people to work remote at this time. When you have to work remote, people have to work remote, even academics move to Zoom, technical health move, and also even government assistance. This is called, which is such as farmers. If this is broadband infrastructure is done correctly, it causes people to have wireless fidelity installed in their homes via router or even through fiber optics. This can also cause for people that do not live close to a hospital to meet with someone over Zoom or even through text messages. Academically, it could impact people to enriching their enhancement to achieve a way to grow academically and scholarly. Thank you.
Okay, so for our final pre-3MT presenter, we have Ray Celeste Tanner from Communication, who will be telling us about Beyond the Org Chart, Informal Communication Network, and Organizational Culture.
When I ask people about organizations that they're a part of, they're always really excited to talk to me about culture. So if they really loved the last place they worked at, they'll say, oh, the culture was amazing. Or if they hated it, oh, the culture was so bad. But what do we mean when we say culture? Well, for someone who studies communication quantitatively, for me, I like to use the competing values framework. This is a theory that was developed by Quinn and Rohrbach in 1981, and it has two main purposes. So if you look at the screen behind me, it identifies 16 different types of organizational effectiveness. So things from morale to training and development. And then it charts them on this graph with structure and with focus. So an organization can really focus on its members or its organizational goals, but it can't be 100% on either one. And an organization can have a lot of structure, or it can be really flexible, but it probably can't be both ends of the spectrum. So where does this come into culture? Well, according to the competing values framework, these types of organizational effectiveness are associated with 4 different culture types, which are seen behind me, from clan, hierarchical, adhocracy, and market. So we know that different types of cultures are associated with different types of organizational effectiveness. And there's a lot of research about ways that you can change the organizational effectiveness. But there's not a lot of research about what contributes to that culture, according to the competing values framework. So my interest is in organizational structures, specifically communication structures. Who talks with who at organizations? Is this department working really closely with another department? Is the relationship reciprocated, or is one department doing a little bit more work than the other? How do these communication structures relate to the different cultures? Because I think that if we can change the structure of our communication networks, we can change our cultures. Thank you.
So we have a People's Choice Award in each track, and now is your chance to vote for the People's Choice Award winner in the Pre-3MT Showcase. So if you scan that QR code, if you're able to, you should be sent to a site, a Mentimeter, where you can vote. Also, you can type in the website and put in the code, or if you're really patient, you can type in the bit.ly. About 30 more seconds to get your votes in. All right. Okay, great. So we're going to move on now to our second track, and that is the official 3-minute thesis competition. So first up, we have Anita Silwall from Communication, who is going to talk about a socio-ecological approach to understand continued use of fentanyl. Anita?
For the past few years, we all have witnessed how our communities have been ravaged by opioid overdose death. Despite several interventions which are based on evidence-based practices, we witness over 150 daily deaths from overdose, contributed by the synthetic opioids like fentanyl in the United States. And do you know that only 2 milligrams of fentanyl that can fit on the tip of the pencil can be fatal? It's 50 times stronger than heroin. In my study, I examined the multi-dimensional aspects embedded within this larger system, recognizing the fact that health and behavior is not just influenced by personal characteristics, but also by social and environmental factors. Using the socio-ecological approach, I examined the intricacies and interplay of these 4 different factors to understand how these factors influence the continued use of fentanyl among those who are taking it or using it. I analyzed in-depth interviews ranging from 30 minutes to 1 hour of 20 individuals, 10 male, 10 female. Among them, 6 of them overdosed between 1 to 11 times. They came back to life up to 11 times. Right? And 60% of them had been or have had a family member who were in jails. What I found was individual factors where people have high level of stress, where they have where they have weaker emotional support, as well as a very traumatic childhood were the contributing factors, whereas family history of substance use, lack of family support and love when they needed, and easy accessibility to substances during their early years were the contributing factors. Moving on to the interpersonal, their family history of substance use were the contributing interpersonal factors. Similarly, I found that the societal factors were the need of the criminal justice reform system, as well as the need to reintegrate and reform the policies. They suggested the need of healing-centered approach to address this issue and a need of family intervention campaigns. And there's no one solution to this issue, and all of us can play an important role in addressing this. Thank you very much.
Okay, next up we have Jovita Dara Ezianwa from chemistry, and she is going to tell us about the $1 million question. How close are we to a cure? Jovita.
Here is the million-dollar question. How close are we to a cure? You see, this brilliant question was from my Lyft driver, John. After he found out I was a UK grad student, he went on to tell me about his wife, Laura, who he lost to lung cancer after a tough battle with treatment. I could clearly see John's pain. Our conversation became a meeting point for the pain of cancer patients and the problem of cancer research. He looked at me for reassurance that what took his wife away will soon go away. But I looked away. I couldn't give him that because I was clearly aware of some of the challenges of current cancer treatment, like 1, resistance. Some of the cures that work today will stop working tomorrow because cancer have found ways to counter them. 2, reoccurrence. Even the people who have been cured today might still relapse in the future. But there's hope. There's hope. Researchers are working relentlessly to come up with alternative cure. My research focuses on crafting alternative cure using SHAPE2 depletion. SHAPE2 is like a thermostat in our body that regulates a lot of other things. Dysfunctional SHAPE2 has been linked to reoccurrence and resistance. Current management for SHAPE2 problem is inhibition, which is really blocking it off and putting it to sleep. But current research has shown that inhibited SHAPE2 does wake up from this sleep and causes resistance and reoccurrence. That's why we're proposing depletion instead. Depletion is causing SHAPE2 to disappear, eliminating the risks of reoccurrence and resistance. My research strategy is in 3 phases. First, design of this drug using computer software. Secondly, making of these drugs in the lab. I've spent the last few years in the lab making various versions of this drug. Then finally, testing it in cancer cells, lung cancer cells. There are 2 exciting results among many others. First, SHAPE2 does disappear. Then secondly, cancer cells die. The future of this research is to fully understand how this happens and then find ways to make this a potential cure for drug-resistant cancer. I really, really, really want to see John again. This time I will look him in the eye and say to him, there are challenges. Yes, there are challenges. There's resistance and reoccurrence, but I have been working harder, trying to design and come up with an alternative strategy for resistant cancers. I hope John doesn't lose hope. I hope cancer researchers and relatives of cancer patients don't lose hope. But most especially, I hope you and I don't lose hope. I hope we hold on to hope because we're really getting closer to a cure. Thank you.
Judges are going to go for a second.
Thank you. Great job.
All right, so next up, we are going to hear about Silica Body, the Skeleton of Grass, by Sarah Chian from Integrated Plant and Soil Sciences. Sarah.
Thank you. So I went to the Kentucky Horse Park Museum for the very first time the other day. If you've never been, they have a lot of horses' skeletons over there. So horses' skeletons are really important for archaeology. One of them even shows how short horses used to be, literally that short back in time. But I study grasses. In grasses, silica body can be considered as the grass skeleton. Silica body is this structure on the grass leaves that grass formed after they're taking up silicon from the roots. Grass is already smaller enough. We all see grass. However, the silica body are even smaller. They're tinier in size and only 1/5 of how thick your hair are. But in order to look at it, the fastest and easiest way is to burn the grass at a really high temperature. And then under the microscope, you will be able to see this two pictures with the white background. What's even better is the silica bodies are autofluorescent, which means by adding a green filter, you will be able to see this two picture with the same area, just the silica bodies glowing green. So my study looking at two different species of grasses, top fescue and bentgrass, and look how they grow and form silica bodies under different rates. And then under different rates. Okay, what we find was after we adding more silicon to the grasses, they form more silica bodies. Makes sense. Second of all, if you look at these two pictures, different species actually form different shapes of silica bodies. So why is my study important? Well, first of all, silica bodies are considered to make the grass stronger and more stress resistant, which means when that happens to the grasses, if they have more silica bodies, they're less likely to be killed by the football players. And then second of all, just going back to the horse skeletons, silica bodies are also really important for the archaeology, as they can identify the grass species based on their shapes, and also locate it where the plants back in time, and also the fire use. So that's my 3 minutes talking about silica bodies. Thank you.
Okay, so next we have Samane Harat-Tabar from Biomedical Engineering, and she will be telling us about navigating retinopathy of prematurity challenge, protecting little eyes.
Let me ask you a question. How many of you have baby or know someone who has a baby? I think almost everyone, yeah? Do you know how many challenges our baby encounter when they are coming to this world? Today I'm here to talk about one of these challenges, retinopathy of prematurity, in short, ROP, which is an eye disease that affects the lives of thousands of babies all around the world each year. In the United States alone, around 28,000 babies are born premature, and about half of them, 14,000, encounter ROP. Moreover, 400 to 600 of these tiny babies lose their eyesight because of ROP. So these statistics show the importance of ROP research. But the importance of ROP research goes beyond just this number. It's about making life better, and reducing the costly impact on of the blindness on the society. Despite advances of early detection and treatment, ROP is still a leading cause of visual impairment in children. But what if we could find a modifiable risk factor that opens the door to a brighter future for these tiny eyes? In our research in biomedical engineering department, with collaboration with pediatricians in Children's Hospital in University of Kentucky, we take a fresh look at ROP, and we focus on a situation where oxygen level drops below the normal level in these infants. And we monitor 210 babies with high-resolution pulse oximeter, and we collect data from bedside. And finally, after analyzing our result, we find that there is a significant relationship between oxygen level and development of ROP. In a simple term, if we be able to control the oxygen level, we would be able to control the development of ROP. And why is this important? This is important because in our research, we figure out this information will help pediatricians to diagnose ROP in very early stage. And also, it enables them to provide affected families some kind of treatments. And we hope our research helps to have a better world for these tiny babies and help them to work independently and pursue their interests. Thank you.
We are having a bit of technical difficulty with the slide advancing itself. I was not over there trying to troll Samana. Okay, thank you all so much for your patience. As we move forward with our 3MT competitors, up next we have Hadi Sabra from Microbiology, Immunology, and Molecular Genetics, who will be talking to us about uncovering a new mechanism to treat Alzheimer's disease.
So Alzheimer's disease is one of those diseases that we hear many stories about, whether it's through a family member, as Alzheimer's disease, there's still no cure for it, there's still no treatment for it. And this is going to be our topic today. Through our research, we have uncovered a new mechanism to treat Alzheimer's disease. So what is Alzheimer's disease? It is a progressive neurodegenerative disorder, and it's the most common cause of dementia in older adults. And it usually poses a substantial economic and emotional burden on the individuals, families, and the healthcare providers. And here in Kentucky, as the population continues to grow, the prevalence of Alzheimer's disease is expected to increase as well. That is why the state must allocate resources for healthcare providers, for support services, and of course, research in order to find a solution. So through research, over 100 genes so far have been linked to Alzheimer's disease. However, we don't particularly understand why or how that association has been established. Well, our lab has studied one of these genes, PILRA, and not only understood its So PILRA normally is associated with an increased risk to develop Alzheimer's disease. However, a synonymous mutation in PILRA is going to be protective, decreasing the risk to develop Alzheimer's disease. And what is a synonymous variant? A synonymous variant is a genetic mutation that is going to change the DNA sequence without altering the final protein product. So if it doesn't alter the final protein product, that begs the question, how is it going to be protective? Well, our lab has found out that this synonymous variant actually affects a sequence at the beginning of the PILRA gene that regulates protein synthesis. So, under normal conditions, when the mutation is not there, we're going to have a normal production of the proteins that are associated with Alzheimer's disease. However, when the mutation is there, we're going to have a decreased production of those proteins, and that's how it's protective. Our lab is the first to show that this kind of genetic mutation, a synonymous variant, is going to be protective by decreasing by affecting the sequence that regulates protein synthesis. And this synonymous variance in research is overlooked, and that is because it does not affect protein product. However, here we showcase its importance. And this research really highlights the importance of these kind of genetic mutations, and it's going to help us further understand the genetic associations, not just with Alzheimer's disease, but so many other diseases as well. Thank you.
Okay, so moving right along, we have Grade Umweki from chemistry, who's going to be telling us how and why the future energy is trash.
Have you ever imagined your food and kitchen waste being as valuable as petroleum-derived fuel? Anyone? Okay. In our lab, that's what we're interested in. We're interested in the conversion of fats, oils, and greases into valuable chemicals and fuel. So at this point, it is safe to say I work with trash. I work specifically with oil waste from fast food restaurants, and I convert them into jet fuels for airplanes. The aviation industry accounts for about 2% of the global greenhouse gas emission, which is a thing of concern to us because in the US alone, the transportation sector is responsible for about 35% of that energy usage. As a result, our lab is currently interested in the conversion of oil waste into transportation fuels, of which we've successfully designed a material, which I want you to think of it as a sponge, or more specifically, SpongeBob with pores and openings, where the magic takes place. So our goal is to fill up these pores and openings with active, inexpensive, and earth-abundant metals such as iron, nickel, and copper, so that when the waste feed passes through these pores, it comes out as 4 different components which are identical to the components you would find in jet fuels, which includes normal alkane, isoalkane, cycloalkanes, and aromatics. For us, we're very much interested in the cycloalkanes and aromatics because these components are responsible for the sea-swell properties of the fuel. That is, these components help maintain the sea-swell integrity of the O-rings found in the aviation fueling systems, which address issues of leaks and other disasters while airborne. Finally, we carried out a techno-economic and a life cycle assessment of this technology, and we found out that this technology is relatively cheap compared to the existing way of making jet fuel. And most importantly, this technology is capable of reducing greenhouse gas emission of about 76%. 76% greenhouse gas emission reduction. That's a lot, and that's what we're proud of. Therefore, knowing how valuable your trash can be, I urge you to go back home, take your garbage to the bank, because the future energy is trash. Thank you.
Okay, our final 3MT competitor tonight is Mary Wallace from entomology, who will be talking about silencing beetle senses, protecting our forests using gene silencing.
In today's rapidly changing landscape, climate extremes are becoming the new normal. Increasingly severe droughts and storms have made our once resilient southern pine forest increasingly susceptible to widespread damage by insect pests, including bark beetles. Now, the beetle at the center of my research, Ips calligraphus, takes advantage of these disturbance events to reach massive outbreak proportions that can result in the death of thousands of acres of trees. Now, this releases stored carbon back into the atmosphere, creating this climate feedback loop. However, it hasn't always been this way. Healthy trees are able to ward off small numbers of bark beetles using their natural defenses. And so, under normal climatic conditions, these beetles preferentially attack stressed or dying trees. Now, this promotes decomposition and returns nutrients to the soil, while also allowing sunlight to reach young trees and foster new growth. So the question emerges of how do we begin to restore the environmental balance between Ips beetles and their pine hosts in the face of climate change? Well, recent breakthroughs in biotechnology have allowed us innovative new pest management tools, including gene silencing. Now, in this approach, an RNA molecule is engineered to turn off the functioning of a specific gene in a target insect. And it does this in a way that's highly species specific,
making it safer than chemical insecticides, even for other insects like pollinators. Now, my previous work has demonstrated that gene silencing can be used to cause mortality in the beetle. However, now I want to refine how we're able to use this technology. Instead of eliminating this potentially beneficial insect from its native environment, what if instead we could eliminate its sense of smell? Now, these beetles rely on complex chemical cues in order to find host trees, to find mates, and most importantly, to overwhelm the host defenses by initiating mass attacks on healthy trees. So, if this process relies on this process relies on a specific component within the beetle's genome, and using an RNA molecule, an RNA molecule that I've designed is able to silence this gene in the insect and therefore hopefully stop the beetle's sense of smell. Now, if we were able to disrupt the beetle's sense of smell, this could open up exciting new avenues of research into how we could protect our trees. Now, preventing effective beetle communication could stop them from reaching these outbreak populations and instead return them to attacking unhealthy trees. Now, as climate change begins to make maintaining healthy forests increasingly difficult, technologies like gene silencing offer a renewed sense of hope in protecting our forest for future generations to come. Thank you.
You picked it right back up. You were great.
It is that time once again to vote for the People's Choice Award winner in the 3MT track. So similar to last time, or the same as last time, you can scan the QR code, use the code on the website, or put in the bit.ly link. So we are going to take some time to do that before we move on to our final track, which is the postdoctoral research showcase. All right, 30 more seconds, and we will move on to our final showcase. Okay, so next is the postdoctoral research showcase, and up first we have Dr. Ajoy Aloysius from biology, who is going to be talking about local heroes of tissue regeneration.
Hello everyone. Let's start by imagining a world where we all possess a superpower like Deadpool. We can regenerate any lost body parts. It's an incredible idea, right? Unfortunately, like many mammals,
humans are quite poor regenerators. We can't even regrow as simple as our fingertip. So the big question here is like, why humans can't regenerate? Interestingly, there are a very few mammalian species like spiny mice have enhanced regeneration capacity in their multiple tissues. Studying those animals will help us to understand what change in our species which make us a poor regenerators. And I am very fortunate to study the regeneration in spiny mice. In my research, I found that there are special immune cells called as tissue-resident macrophages, which acts like a first responders in the tissue injury of the spiny mice. So to make it much more clear, I want to use an analogy here. Let's imagine a healthy, normal tissue as like a smooth flowing traffic. Everything is going well. But injury is like an accident. Then what happens? Vehicle collides, people scream, and everything come in halt. Who come to your rescue first? Yes, the first responders, mostly the local policeman in that area. Here, the tissue-resident macrophages in the healthy tissue act like a policeman. When the injury happens, they swiftly move to the injury site, and as just policeman securing the accident site, the macrophages also securing the injury site and also not allowing any external threats as well as the other immune cells. Later, they're helping for rebuilding the tissue. And when I remove the resident macrophages, the spiny mice lost its tissue regeneration capacity completely. That suggests that they're very important for the regeneration. So when we go back to the analogy, in the absence of policemen, what happens? The small issue can be taken care of by other external forces. Most often, they ends up in escalation of the issue, and eventually it's like a chaos. Similarly here, when I remove the resident macrophages, the infiltrating cells from the blood come and then make it more worse, and high inflammation ends up in scarring. So the take-home message here is, as like the local policemen, the tissue-resident macrophages are the local heroes of regeneration. And at one day, studying all those things will make us like a superhuman power which we can regenerate all. Thank you.
Okay, our final presenter tonight is Kine Ong from Public Health, and Dr. Kine Ong from Public Health, and she will be talking about Alzheimer's disease, a polygenic risk score analysis.
Hello everyone. Today I would like to talk about polygenic risk score analysis to detect the Alzheimer's disease. Do you know how many Americans are suffering from the Alzheimer's disease? More than 6 million of Americans are living with the Alzheimer's disease nowadays, and the number is expected to begin double by the year of 2015. So in this case, we need to detect the Alzheimer's disease as early as possible. So what can we do? Unfortunately, Alzheimer's disease is caused by 80% genetic, and then DNA variation from the gene called ApoE is important for the late onset of Alzheimer's disease, but that can only explain 10% of the risk of developing the disease. And scientists up to date, they discover 18 common DNA variations throughout the genome of our body. But that can only explain 40% of the risk of developing the Alzheimer's disease. So what about the rest of the risk? What will you do as a medical scientist or medical researcher? So in this case, the role polygenic risk score became crucial. You may think, what is she talking about? What are the polygenic risk score? We don't know. So I will explain in very simple word. So polygenic risk score is a single measure that combines the risk of developing Alzheimer's disease from millions or probably billions of genetic variation through the body, and they combine a risk. It is setting like a risk threshold. So we sort the people according to the threshold, and the people who have high polygenic risk score are more likely to have the disease. So in this case, we can get benefits by early intervention and early prevention from getting not getting the Alzheimer's disease. So my analysis now is focusing on the developing the Alzheimer's disease risk score to give the individual risks. I believe the result from this study can give the benefit to everybody to know the Alzheimer's disease as early as possible. And I also hope to contribute to the precision medicine for each and every individual. So stay tuned. Thank you.
Okay, this is the final People's Choice Award vote for the postdoctoral showcase. Same instructions apply here. And while we are doing our final People's Choice, I believe the judges are going to gather and debrief. And let's go ahead and give another round of applause for all of our competitors. While the judges are debriefing, we will come back to announce the winners. So stick around. Okay, so I don't know about you all, but I was very inspired by all of the great research that our graduate students and postdocs are doing on campus, and also their courage and bravery in standing up here and presenting that research in just 3 minutes. So again, round of applause.
Tell them right now to stay for photos.
Oh yeah. For the presenters, if everyone can stay for photos, we are going to take a series of photos of you all. Okay, so for our winners, in the pre-3MT track, third place we have Devin Dunnigan. Second place, Hannah Blevins. First place, Ray Celeste Tanner. And Ray Celeste Tanner is also our People's Choice Award winner. And I should mention that these awards come with a monetary value or monetary award. So all of our winners will be receiving that in your UK accounts after the event. We don't have big checks tonight, so. Okay, so we'll go ahead and go to our 3MT round. Third place, we have Mary Wallace. Second place, Great Umweki. And first place, Jovita Darizianwa. Jovita is also our People's Choice Award winner. And for winning first place, Jovita will get to compete in the regionals for the Council of Grad Schools. Okay, for our postdoctoral showcase, first place we have Dr. Kai Nyung. Second place, Ajoy Aloysius. And Dr. Kai Nyung is also our People's Choice Award winner. Thank you all so much for coming out tonight, supporting the graduate school, our graduate students, and our postdocs. And stay for pictures.